everyone.
Hey, y'all.
Howdy.
Well, thank you guys. I'm in the background. I'm just a floating voice over here, but my name is Courtney Everhart. I work in the marketing department, and I am so appreciative of all the questions that have been submitted. But I've got some experts here who are way better at answering those questions. So we've got Katy Rowe-Schurwanz and Janine Cloud, and here to answer your questions. So feel free if you want to, add any questions in the comments, feel free to do so. But we are going to go ahead and answer all of the questions that we received in our form first. So to get us started, let's see, we've got some good ones in here. We're going to start with these from Lily. Okay, so what is the difference between your Y-DNA test? What's the difference between Y37, Y111, and the big Y700?
So the bigger the number at the end, the more of the Y chromosome it's testing. And the more of the Y chromosome that you test, then the more information you can find out about your lineage. So you get more detailed information about matches and when you share a common ancestor with them. You'll get more detailed information about your placement on the haplotree, which is the genetic family tree of humankind.
I always like to use the analogy of a puzzle when it comes to STRs. It's like if you have a giant puzzle, the number of markers that you're looking at, it's like you're looking at that portion of the picture. So if you have a puzzle that has, say, 111 pieces, if you're looking at 37 of them, you're getting part of the picture. It's going to be accurate in terms of that part of the picture that you're looking at, and you can extrapolate the rest of the picture from that. But by testing the extra markers, it actually fills that in, and it gives you a better idea if some of the predictions that you were making were correct, or if maybe you need to shift your focus a little bit on it. So any level that you're at is good, because if you have someone who you're thinking you might match with, you're thinking that they might be a good resource for you for your genealogical goals, you can always start with that smaller portion of the picture. And if that jives with what you're looking at and what you expect, then you can go from there and expand the data that you have.
Very nice.
I'm going to steal that puzzle analogy from you, Jen.
So another question we have is, will the Y111 update my haplogroup?
No. So with the Y37 and the Y111, you're getting a predicted haplogroup. It's going to put you at the top of your branch on the haplotree, the genetic family tree of humankind. With the Family Finder, you can get a bit more specific. So you'll get a little further down on the branch, and then the big Y gives you the most specific placement. So your haplogroup will stay the same predicted one. If you go from 37 to 111, it'll get a little better if you've got a Family Finder, and it'll be the best if you've got the big Y700.
And I think it's important to understand that with Y-DNA and mtDNA, unlike autosomal where you're starting with yourself and going backwards in time, with Y and mtDNA, you're starting in the distant past and bringing that forward in time to where we are now. And the more information that you have, the more, the closer that you can bring that to where you are now.
If I could jump in and just explain, because this is a question I get a lot of, what exactly is a predicted haplogroup? So STR markers, they're long stretches of DNA. There are areas in DNA where the same sequence of DNA will repeat itself over and over. And we count the number of repeats at that location, and that's what we consider to be the value of that. So when people share the same values, it's an indication that they share the same ancestor who passed those values down to them. But the problem is that STRs, relatively speaking, mutate faster than SNPs do. So they're able to give you a good idea of where they are. And so that's where the puzzle analogy comes in, because the more STR markers that you have, the more it's able to give you a clear indication. But to be able to pin down specifically where you are on the tree, specifically which branch you are, we use SNPs because they're much more stable and they're much more specific. So the way that STR predictions work, or DNA predictions, is we have a database of people who have taken both STR tests and SNP tests. So we're able to get sort of a signature of what those STRs look like. So we can say, oh, well, for this particular SNP or for this particular branch of the tree, we tend to see this range of values in STRs. So when you take an STR test, it will compare those values that you have and be able to predict that, okay, well, if you match the same values that all these other people have that have this SNP, then it's a good indication that you have that SNP as well. So the predictions, they tend to be older, they tend to be further upstream on the tree because there could be a lot of variation within that predicted haplogroup. So it doesn't want to specify that you're on one branch when you might be on a slightly different branch. So a SNP test is able to pinpoint down where it is. So the Family Finder test, that's it. That is the Y-DNA that's on that. Those are SNP based. So that's able to give you a confirmed haplogroup, but it's limited in the number of SNPs that chip tests for. So that's why, like Katy said, it gives you sort of a mid-range level there of a haplogroup. So you have predicted haplogroups based on STRs. Those are really high level. Then you have confirmed haplogroups with Family Finder. Those are mid-level because they're a set of pre-programmed SNPs that are in it. And then you have the big Y, which is, it looks at an entire region that has a lot of SNPs. So it's able to discover new SNPs, and it's able to find the finer points in those branches to where you belong. So a lot of times people ask, okay, well, if I upgrade the level of STR tests that I have, is that going to refine my haplogroup? Well, no, because again, you're just getting the prediction from the STR. So it takes the SNP test to be able to pinpoint where it is. So it's always a good idea. It is an idea to start with the lower level test to see if that test candidate warrants upgrading to the higher level. So there's a few different ways that you can look at it, and it depends on the resolution that you want.
Thank you, Jim. Actually, that kind of is a great segue into another question that somebody submitted. What is a terminal SNP?
It's a SNP that doesn't have very long.
No, it means like your final placement on the haplotree. It's not a term that we really use anymore. And part of that is because with more testers coming along, your placement can change. So the more testers you have, the more specific your haplogroup can potentially get, because we may be discovering new SNPs on that branch.
A term we generally like to use instead is furthest downstream, because as the haplotree is today, that is the furthest point. It's the terminal point. But as new SNPs are discovered and they're added to the tree, that terminal position could move. So terminal is a bit of a misnomer. It's just the highest resolution that we currently have based on the database.
Now, I will say that when you take big Y, there comes a point where you don't have any more private variants, meaning you don't the sequenced test has not discovered anything else that's unique that nobody else yet has. Then that portion of your test isn't going to move very much. But the branches can move based on other people testing. So in big Y, it's different from the other tests because it's not going in and saying, okay, we're looking for this one, this one, this one, and this one. We're not looking for specific SNPs like in Family Finder, that they choose specific ones to make sure that we cover all the haplogroups. Well, big Y is looking for whatever's there for whatever you have. And that's why it's called a sequence test. It goes through those. So that one gives you that more refined, because it's not only looking for program stuff, it's looking for for what you have. And once you get to that point where you don't have any more, you've matched up with everybody. Say you tested your uncle or your brother or whatever, who's very close to you. So you have your branch, it's only that branch itself that's going to move, not your particular part of it.
When I first took a Big Y test in 2015 or 2016, I had somewhere around 30 private variants that I was the only person in the database who had them. Today, I have one. I have one private variant. That means it's moved 29 times further down on the tree. So again, that's why if I had said 10 years ago, oh, well, I already have my terminal SNP, like, oh, past me. Oh.
and that whole explanation there helps you understand why, the more people who test and the more data points you have, the more that the probability algorithms can determine those times to most recent common ancestor. For example, my uncle and my brother tested, and theirs is really very recent. It's actually like 1888, which is when my grandfather was born. But if you have, there's nobody on a branch near you for 500 years, it's kind of hard to pinpoint that date. So it's all about more data points.
Yeah, whenever I get I have some people who will ask, like, well, there's not a lot of people. I looked up my surname in your database and there's not a lot of people, so I'm not sure if I'm going to get anything from it. But the thing is that a rising tide raises all ships. That if everyone has the mentality of, well, there aren't other people with my surname, so I'm not going to test, then no one with your surname is going to be there. So as more people test, as you get your cousins, your uncles tested, then it's able to increase that data point, and that's able to help not only your branch, but the entire tree when it comes to those time to most recent common ancestors.
Plus, your matches may not have your surname, because surnames are a fairly recent evolvement with Western culture especially. And so your matches may have other surnames, there can be other reasons. For example, my brother has his stepfather's surname instead of my dad's surname. So they're different. So if you go in just looking for surnames and not genetic matches, then you're already limiting yourself out of the box. So that's something to consider.
Mm-hmm. So we've got some comments that are coming in, some pretty good questions, all relating to the conversation that we've been having. So I'm going to start kind of reading those off so they don't get lost. But Shelley wants to know, why is the birth date range for the most recent common ancestor in the Big Y time chart so big?
It'll vary based on which part of the tree you're looking at. So the more testers in a part of the tree, the more recent that time the most recent common ancestor age range is going to be.
So it'll be a smaller.
Yeah.
It narrows.
It narrows it down.
Okay.
Yeah. If there's not very many testers there, then it could be from here.
It's got bigger data points. Okay.
And that's something else I've noticed in my own tree is I've seen some of those time ranges shrink.
Mm-hmm.
Perfect. Well, hopefully, Shelley, that answers your question. Eleanor has a question. How do you know which SNPs to test?
It's very difficult to just pick SNPs. There's 22 million SNPs at the Big Y test. So just grabbing a random SNP out of the box there isn't really going to help you. So that's why with the Big Y, we scan all of that information in the lab. We scan all of that. And we see what results you have for all of those SNPs. You may not get an answer for all of them, but you'll get an answer for a lot of them. And we can discover new ones too that we didn't know existed.
So I think also, go ahead, Jim. Sorry.
Well, just along those lines of what SNPs to test, in the early days of the Big Y, as we were discovering new branches, we did tailor, we call them SNP packs. They were for specific branches to, if you had a prediction for a branch, then you could take a SNP pack and it would refine that branch. But the Big Y has produced so much data, it's produced so many tens of thousands of SNPs that those SNP packs quickly became obsolete because, they took you pretty far down as the tree was at the time the SNP pack was made. But it's increased a lot since then. So that's why we really encourage some of these more exploratory tests like the Big Y, because it can pick up not only new SNPs, but it's able to look at that entire branch and all the things that could be in it, as opposed to a targeted panel, which is limited in its scope.
Now the targeted panel is sort of migrated over to Family Finder. Family Finder uses SNPs to match you with autosomal DNA, but it also includes Y-DNA SNPs. It's an mtDNA SNPs in it. We currently provide that Y haplogroup based on that, and those SNPs were chosen specifically for that mid-range to be able to give that targeted, to hit the most targets, but still be downstream from that predicted haplogroup. So it gives you a more specific. It gives you the county instead of just the country, but it doesn't give you the city and the street.
That's a good analogy. I want to say, just from when Eleanor's comment came up, that she was also asking, so how do you know which SNPs to test on Family Finder? We talk about SNPs on Family Finder. How do we know which ones to look for?
That's where our population geneticists get the big bucks.
That's our research and development folks are determining that information.
And one of the things that they do have to consider is that because we do take transfers from other companies, they have to have some overlap with the other companies so that they can be made compatible.
And then I've got one here, and this is going back to YDNA, and this is from Cat M934, and I'm just going to read it off. I don't fully understand it, but I'm hoping y'all will. It says, what does it mean when you have non-matching variants that are already named on the tree in unrelated haplogroups and aren't shared by the members in your exact haplogroup?
So some of your non-matching variants, there are some SNPs on the YDNA haplotree or in YDNA that have a name, but they're not phylogenetically relevant. So they're not used for building the tree. They're not used for placing you in a haplogroup. And you might match someone on that. You might not match someone on that. These are usually SNPs that are in like a messy area that's hard to read.
Well, plus you get a big chunk of the Y chromosome, and a lot of people have that chunk in common. It's only when you get to the part that's in your branch that you see those unique SNPs or those things that match your haplogroup and your relatives. Like some of the SNPs, they go back along that trail back into to Africa. So those SNPs are going to be there. You're going to be positive for them down that branch, but just because one of your people in your close group doesn't have it, it isn't particularly relevant. The ones that you're focusing on are that breadcrumb that's across the top of the block tree, or that's on on one of the Discover reports down the time tree. Those are the ones that are relevant to your branch.
And to clarify what we mean by phylogenetically relevant, because that's sort of an umbrella term that could apply to a lot of different things. And so the question you were asking about, like, well, you see it on the tree, but you don't see it on your particular branch. There are some SNPs, they're called recurrent SNPs, meaning that they appear independently in multiple haplogroups. So it could be that if like, let's say you're an R1b, and there is a SNP, it's a really ancient one that everyone in R1b has. Well, it's not phylogenetically relevant because it doesn't narrow anything down. But if that same SNP occurs in, let's say, haplogroup L or haplogroup E, but it occurred independently more recently, meaning that it's shared by some people in that haplogroup, but not all of them, then it is phylogenetically relevant. So that means it could be a named SNP that is on the tree, but we only put it on the branch of the tree where it's actually meaningful. So that's why you could have a named SNP in a non-matching variant, or a named SNP that isn't appearing on your tree for you because it's not listed in your branch of the tree.
It could also be, something that'll eventually change the formation of the tree as well. You can end up splitting a branch as well. Like, it could be a SNP that we discovered a long time ago, but now because more testers are coming up, some people are testing positive for it, some people are testing negative for it, and so our our scientists can actually place that in a better place, a better branch on the tree based on the new information that's coming in.
Or it may be from an ancient sample that we get that gives structure of the tree from hundreds of or thousands of years ago, because we have new information from an old source. And I'm wondering if Kat is talking about Y-DNA or mtDNA.
Kat, if you don't mind, because he or she has been commenting. So go ahead and let us know if it's Y-DNA or mtDNA.
We've been answering Y-DNA.
Yeah, we have been focusing on Y-DNA. One thing that we did have a question. So why doesn't, we're going to kind of shift. This is a great shift into autosomal DNA, but why doesn't Y-DNA show ethnicity percentages like autosomal DNA does?
Because you can't get a percentage out of one.
Well, that was easy.
Y-DNA is one single line, so you can't get a percentage out of it. Your autosomal DNA gives you 100% of your DNA, so you can break that down into percentages. You're getting contributions from all of your ancestors, but if it's only one ancestor contributing, you get a source, you get a path, a migratory path, but you can't get a percentage because it's just one.
Yeah, the migratory path is because that one SNP represents one ancestor who lived in one place, and then their paternal ancestor, potentially migrated from another place. So you're able to follow that one specific trail of breadcrumbs. And when you do that, it creates a geographic path rather than a pattern, because like Janine said, or rather than a percentage, because like Janine said, the percentage is determined from all your different ancestors, not just the one Y-DNA ancestor.
So on that same note, we also had another question that was submitted to us. Is it possible to provide a basic diagnostic tool that includes Family Finder with one of the STR marker tests like Y12 or Y37?
We don't sell the Y12 test anymore, but we do have the Y37 and the Y111 and the Big Y available to bundle with on our homepage. You can go to shop and then bundles, and you can see all of those there. You can even add an mtDNA if you like.
And you generally get a discount. There's a discount for bundling the tests together, so it is cheaper to buy them at the same time.
That's true. That's a good point. So
really fast on those lines, I don't know if I can share my screen, but we do have a tool that's called Advanced Matches. So if you've taken multiple types of tests, if you scroll all the way down to the bottom of your dashboard, there's a tool that's called Advanced Matches. And what you can do with this is you can select multiple different test types to be able to see if they match on all of them. So that can be a really powerful tool, because autosomal matches, it can be hard to tell what branch of the family that they're on. But you know that they're probably going to be fairly recent in time, at least relatively speaking. So you could compare your matches through an autosomal test as well as a Y test. And if they match you on both, then you're able to know that it's fairly recent and which line you potentially match them on.
Love it. So we have kind of circled around mtDNA. I have a question that I've seen come up a lot. We updated the mtDNA tree earlier this year. We added 35,000 branches, and then in November we added an additional 12,700 branches. What does that mean?
Those are more ancestors on the family tree. Each one of those branches, each SNP, whether it's Y-DNA or mtDNA, represents an ancestor. So the more ancestors you have in your family tree, the more complete it's going to get. Okay.
Yeah, the names of SNPs can be really overwhelming to people because it's this long stream of letters and numbers. Basically, that's the designation that we have for the specific mutation of the specific location on it. But that mutation happened in an individual. We just maybe don't know the name of that individual. So we just designate that SNP, but that SNP came from a person. So if you look at the haplotree as a family tree of people that we don't have the names for, then to me, at least as a genealogist, it makes a little more sense.
it's like a huge pedigree chart where the mtDNA haplotree is your pedigree, your maternal pedigree, and the Y is your paternal pedigree. And some of the cousins are more distantly related than others.
So why are there, Y DNA haplogroups are very straightforward. You've got letters and you've got numbers. Why in mtDNA haplogroups do you have random symbols like a caret or an exclamation point?
We've got an article on that in our help center, and it's been a while since I read it.
Well, here's the thing. The first thing you have to remember is that Y DNA, big Y covers 16 to 22 million base pairs, of the Y chromosome. The mtDNA genome is only 569 base pairs. So it's very small, comparatively speaking. So the terminology has to be totally different because you're looking at a totally different structure. And so with SNPs, they're for Y DNA, they're at location. And the SNP is single nucleotide polymorphism, which is a fancy way of saying that it changes from one base to another base at a single location on the chromosome. Well, with mtDNA, it actually lists that change. Like the first one, it will be whatever base it was, whatever. And then that 16,569, wherever that location is, and then the letter that it changes to. So it's a lot more specific. It is kind of confusing though, because those parts of that address can be in different haplogroups. And so it takes a lot to figure it out, which is why it took so long for them to, add those branches on it. But yeah, that's
another reason for that too is that within your cell, you have the nucleus, and that has your nuclear DNA, your Y DNA, your autosomal DNA. You just have it's all contained there. But your mitochondria DNA, it's in the mitochondria, and that's found throughout the cell. And whenever a mutation happens in the mitochondria, it doesn't necessarily happen in all the mitochondria in a cell. It could happen in some of those. So the mutations, they tend to be a little more gradual over time. So some of the different symbols that are in there, those indicate various, I guess you could, in a very simple way, you could say various stages of mutation that they're in. It can also indicate sometimes where it has changed from one value to another and then back to the original one. And we call that a back mutation. And we know that because of its placement in the tree, and we see where all of the other SNPs are. So if we're able to see all of those other mutations, we're able to know where that location should be. And based on the value, we're able to get a good idea of, did it change? Did it change back? Is it in the process of changing, etc.? And like Katie said, there is an article in there. I don't know if I'd read it. It's written by some guy named Jim Brewster. He's
and I also want to add Roberta Estes, who is one of the builders of our new mito tree, added a comment here that part of it was they're trying to keep compatibility with the previous haplogroups that were on the old phyllo tree, and so the naming structure got a little more complex because of that.
Love it. I'm glad that you called out Roberta, because I was going to call her out myself. So she's lurking in the comments. She's watching us. We love us some Roberta. Okay, so let's see. One question that we got was actually from one of our group project administrators. And I know, we've got some people here who are customers of ours and who aren't. So before I jump into this, do y'all mind going over what group projects are?
Oh, you take that.
Me? Why me? I'm actually have been group project manager for a number of years now, so that's kind of been my focus with the company, is working with group projects. And what they are is simply a way for people to get together and share common goals in their research, whether it be as a surname, or if it's a geographic region, haplogroup. Each one of them has a different function and allows you to compare and share information and do more targeted research in that particular area. Did I miss something?
It's a team for your research.
Love it. Okay, so with that, one of the admins has written in, and they were just wondering if there are any tips on helping to persuade or to educate their distant cousins that they want to test, but might be hesitant, not just because of price point. Maybe there's other reasons. So is there anything that we can tell them, any advice that we can give?
So there's a lot of ways to do it, and I'll let you go over some of those. But one thing I'd say is to, when you're talking to them about it, focus on the discoveries. Like, here's what I've been able to discover so far about our shared history. Here's what I'm looking to discover, and I can do that with your help. And then,
Well, I always think it's important to acknowledge people's concerns, whether it's about privacy or, finding out things that you might not be ready to find out. And then the number one rule is that if you don't really want to know, don't test, because once you've tested, you can't go back. So if somebody really doesn't want to know, then I wouldn't push them. But if they're concerned about privacy, we have our privacy policy and terms of service are linked at the bottom of the page. Our customer service team is always available to help understand that information and know what limitations are there, what protections that we have, what we follow, all of the guidelines and regulations for not just states in the United States, but also Europe. There's a lot of regulations involved. But I can understand that there are concerns about it. You are putting information out there. So, some people use aliases, which is fine. Some people will allow somebody else to manage their kids so that their personal name isn't even involved with it. Those are strategies. If you're going to do that, then there's some back end work that you kind of need to do to make sure that there's no confusion as to who handles what. But there are various ways of encouraging people to test. One of the things that you could do is go through your autosomal matches and target the ones that fit, and don't do a mass email, but make sure that when you're sending emails to people that you include enough information in it that it makes it attractive to them to answer, instead of we match, how do we match? Especially if it's somebody that you're like, hey, you fit in right here. I think you could take me here. I'm willing to pay to find out if you do, that kind of thing. Some people have, like was saying, that they have a scholarship for it. The group projects have what's called the group general fund, where people can donate, and that way it's kind of a company thing. And so it's a little easier to say, well, the project is willing to put X amount of dollars towards this. And the administrators can send out bulk emails to their people. They can invite people to join the project. We have a certain match where they can, any Y tester that's not in the project can be invited, that kind of thing. So there's a number of things that you can do there. And I think we did have a blog post about this a while back. We should probably revisit that in next year and kind of freshen that up for 2026.
And one additional thing we're adding in 2026 as well will be the ability for you to share purchasability with another match or another user. So if you've got someone who wants to pay for your tests, but you don't want to give them any access to your account, which is definitely understandable, rather than having to call up customer support and share emails with customer support, so you can say, yes, I have this permission. If you just go into your account, click a couple of buttons, and then they'll be able to add something to your shopping cart, and that's it.
And I think it's really important as well to emphasize that the DNA and the DNA results belong to the tester, period. End of story. I don't care who paid for it. I don't care if we paid for it. It doesn't matter. It belongs to that person. And so whatever that person wants to do with it, it's completely up to them. So they have, so if if Mary says, I'm going to talk my brother Joe into testing, and he's concerned about this or that. If Joe tests and then he's like, I don't want this in the database anymore, take it out, then we take it out. So there's those kinds of things as well.
And for me, I find that some of my family members just don't really care. And I know that, a lot of you are probably the genealogists for your family. So, you go and talk to Joe, and Joe's like, yeah, it doesn't matter to me. You could be like, well, yeah, but it matters for, our family history. And so I'm willing to take care of it. I just need the sample. And so for me, I manage a bunch of my cousins' kids who have never signed into their own kit because they don't care. But me as an administrator of my own family project, it's very valuable to me. And, so I make them aware that the data still belongs to you, but that data is valuable for the family in general.
And he hits on a good point. Legacy is a big deal. I asked one of my uncles to test for me, and he graciously did. And his sample provided, he wasn't interested per se in any of it. He's, just like, okay, whatever. But because he did that, I now have his Y DNA, his autosomal DNA, and his mitochondrial DNA. So I have that whole, my paternal line. I have my father's mother's line, which turns out to cross my mother's father's line anyway. And then the autosomal as well. So he's left this legacy of information that, he's gone now, unfortunately, but that information that he provided will live on, so that the rest of our branch of the family has that we wouldn't have gotten otherwise. And so appealing to that a lot of times is what they might not particularly care for themselves, but they might be interested in giving that legacy to those who are interested in it.
Yeah, that's a really good point. We're gonna kind of shift gears a bit. Back to YDNA. We got another question about this. Will FamilyTreeDNA release a new, more advanced YDNA ancestry test that is stronger than the current Big Y 700 test?
When the technology is there. It's not there yet.
And it's more than just the technology, but it's you have to have the technology, you have to have the pipeline to deliver it, and it has to be at a price point where people can buy it. And so it has to be a marriage of all of those things to be able to do it. We're all for it. We're all for technology advancing, having the most information. We're all about all that. But the consideration for being able to have it across the database, and enrich the database, is that it has to kind of meet those things, and there's nothing there yet. But
cost is another major factor, because the more of the genome that you look at, the more cost is involved in terms of testing that big stretch of genome. And as technology improves, prices are able to go down. We're able to expand coverage. So, like they said, I'm sure we haven't reached the end, the terminal SNP level of technology yet. So, it's 10 years from now, I'm sure there's going to be more coverage.
Yeah, I'm going to only open this door just a little bit. Whole genome sequencing is there. It's available. It's just that having the data is only one part of it. That's only one small part of it. Big Y was originally supposed to be a data dump. It was supposed to be, here's your data, have fun with it. And here we are,
11.
Because that was in 20, yeah, it was 20 years later. And we have this ginormous haplotree and all that. You have to be able to take that information and do something with it. You have to make it relevant. You have to be able to extract it in ways that are useful and that are financially viable to be able to do them. So, the guys are always looking at stuff. We got dreamers
And I add too, like we got to have ways that are useful for genealogy, because that's what we do. Like a lot of them are useful for medical tests and so what and things like that, but that's not what we do at FamilyTreeDNA. So our lab can offer that as a different thing, gene by gene.
And we have to be careful, because if we say that we're interested in XYZ, then next week it's going to be all over that we're doing X, Y, and Z, when that's not the case. So we have to be very careful about what we say, because it gets amplified fairly quickly.
Yeah.
Another consideration with testing technologies are that different testing platforms sometimes produce different types of data, and those different types of data are in different formats. And so it's not only a matter of having the technology to be able to produce that data, but to be able to build an infrastructure that can read and interpret that data in useful ways. So things like a whole genome sequence test, for example, it produces data in different ways than the big Y does. And so there's a challenge to be able to make that compatible. So that's another factor that goes into it.
Getting it onto the page is always the challenge.
That's a good point.
And keeping it on the page. So.
Yes.
Jim, you had mentioned about other testing platforms, and somebody earlier in our live event had asked, how is it, and I assume this is autosomal, but if Summer Z7U is still with us, if you want to clarify, you can, but how is it a person can take a DNA test with two companies and they have different results?
Each company's got their own database of testers. And from that database of testers, each company's got their team of population geneticists who decide, what populations you're going to cover and give results for. So one company might, give results for, Sephardic Jewish, and another company might not. And depending on how many of those different categories there are, how many different populations there are, and what they are is going to decide what your results are.
Okay.
And another factor with that too is that we've mentioned testing chips before. So for those of you who are wondering what that is, don't worry, they're not Doritos, even nacho flavored ones. No, they basically have receptors for individual markers, for individual autosomal SNPs. But each one, and some of those SNPs are Y-DNA or mtDNA, that's how you can get the haplogroups. But those chips are custom built. So they're very similar from one company to another, but they don't look at exactly the same markers. It's more of a Venn diagram sort of thing, where there's a lot of overlap between what the different testing companies test for, but there's some differences as well. So if the reference populations that a company is using are in part of include some of those markers that we don't test for, or vice versa, then that information just isn't available to be able to compare it to that reference population. So the proprietary differences between what's tested and what is considered a reference population changes from one company to another.
Two quick things with that. One is time frames are also a thing. It could be the same populations, but it could be if the test is looking at a different point in time, then the results could be different for the same people from company to company, and then I forgot what the other one was.
I get it. It happens to me too. But you bring up a really good point. I think I've never thought of that you can be looking at the same geographical population, but in different points of time, their DNA is going to look different.
Yeah,
I've never thought of that. So
right, because it depends on where they were when, kind of thing, and then of course you've got random recombination, and when you're looking at the overall picture of it. Oh, I know what it was. To break it down really simple, if you think about the kid's toy, that's it's like a ball, but it's got holes in it, like a star, a circle, that kind of thing, and you're taking those pieces and you're trying to fit them into the hole. Well, with population geneticists, you're trying to take and fit it into the hole that it fits the closest. It may not be exact. For example, with Native American, I'm Cherokee, and when my Native American comes up, it's always going to be like Mexico and South America, because we don't have a population for Cherokee specifically for North America. So it's kind of like that across the whole spectrum. It may look like this. So, for example, if your ancestors were from northern Scotland, where there was a heavy influx from the Vikings and everything, that then it may look like this one thing when that's the closest that it could fit into. And then there are other populations, like she mentioned, Sephardic DNA. There are different origins because Judaism is a religion, and so there are different geographic points where that comes into play. And so there are at least 3 points where Sephardic DNA comes from, and it depends on which of those samples that you have to compare it to as to whether it matches or not. So like if you have one from Morocco, but your branch came from Turkey, it's not going to necessarily match that Morocco one. It might show up as North Africa instead. Does that make sense?
Yeah.
To go back to what they were saying about time frames with it, to give sort of an analogy that's somewhat similar for Y-DNA for me. One of my ancestors is William Brewster. He was a Mayflower Pilgrim. He was born in England. He and I are in haplogroup I1 or IM253, which is a Scandinavian haplogroup. So you could say, well, my ancestors are from England. Why am I having a Scandinavian haplogroup? Well, that probably indicates an influx of people to England prior to when William Brewster or whoever his first ancestor in England were born. So that migration is the same thing that you happen in any different area. So if you have a reference population of people who have lived in one area for a very long time, eventually, if you go back far enough, they came from somewhere else. So you're going to see some overlap in the DNA, especially when you talk about neighboring regions. And so it's, again, with that Venn diagram, it's going to try to fit you into the region that is best. But because those shared regions also share some of the same DNA, you may find portions of your DNA matching to neighboring populations from what your true ancestral population is. And that's always, and that is another type of technology and science that's always improving, to be able to narrow that down, to be able to get better and more reference populations to give you a more accurate result for it. So that's another reason why you may see it change and why you see differences from one company to another.
Okay. These are all really good. One question that was asked at the very beginning, and I'm pretty sure Cat ZK8BV is still online with us because I've seen a few more comments, but they just received their test kit. Is there anything that affects the results? Diet, medication, etc.?
Say denture glue is the big one.
Well, to start with, you want to make sure that you wait about an hour after eating or drinking any kind of hot beverage, anything that's going to affect or coat the cheek cells. If you're doing, if you have some sort of chemotherapy, radiation, something like that, you might want to wait until just before your next cycle to do it. Unless you're having whole brain radiation, it's not really going to affect it that much, but it's still better to just wait right before that, so you get that good deal. You want to make sure that if you wear dentures or any sort of mouth appliance, that you remove it and make sure your mouth is clean, because one of the main things that causes samples to fail is contamination from non-human sources. That means, bacteria, food particles, things like that you don't realize that are there, but really are. And denture cream actually it dissolves the DNA. It makes it look like you do not have DNA at all. yeah, it's pretty crazy. It took us a long time to figure that one out.
And then two things that could affect it as well would be if you've had a stem cell transplant or a bone marrow transplant.
Yes.
That can change the composition of your DNA.
Or an inside of the mouth skin graft. That's the third thing that is very rare, but it happens.
And another absolutely rare thing that can happen is chimerism. It's not something that you want to just say, oh my God, if you have this result, then you're whatever. No, it's not like that. It's very, very rare that it happens where, some person does have two forms of DNA, within the same person, but it can happen. We see all kinds of interesting things happen.
Yeah, the biggest thing is really just make sure that you have a clean mouth when you're doing it. My wife's grandpa, we got him tested. He has since passed, and so we wanted to get his DNA while we could, and the only chance we had was at a family barbecue. And I did my best to wait until after he finished eating, but I'm pretty sure that one of the two vials was mostly barbecue sauce. I submitted the other one instead.
One thing that I would like to kind of ask and get clarification, since you guys are the experts, but we do average second person. People are allowed to come to our booth and they can swab right then and there. We have a whole swab station. Mm-hmm. But a lot of times I end up telling people, just wait even to drink water, because I would rather you have nothing in your mouth, just have it clean. Don't even, if you've already eaten and drinking whatever, then do it first thing in the morning before you even brush your teeth. Is that overkill?
Oh, yeah.
Okay, well, that's on brand for me, but
yeah.
Okay.
The water's especially fine because it helps reduce some of that additional bacteria.
Okay.
But I cornered one of my cousins after dinner, after Thanksgiving dinner, and said, "Here," and there was probably coffee on it, but it was fine. And you just make sure that you swab. They want to really swab. And here's another thing that sounds like it might be a no-brainer, but the actual tip of the swab needs to go in the vial. You don't just dip it in there, because that's not how it works. You have to break it off and put the swab in there. We have them come in all the time with no swabs in them because the person thought they just dip it in there. So
also, we would like to note that we only test humans, so
yes,
that's another caveat.
We had one person submit dog DNA, and then we wrote them and we're like, yeah, this isn't human DNA. He's like, oh yeah, I swabbed my dog because I wanted to see if you could tell or not. So yes, we can tell the difference.
We could tell that it was not human.
Yes.
But I want to make sure this is very clear. We can't tell how old you are by your DNA. We can tell male from female. We can tell, there's a lot of factors in there that I won't get into, but let's just say that there's a whole lot that goes on beneath the surface that you're not going to know unless you have a genetic test. So yeah, but we can't tell whether the person you tested was a child, adult, old person, or anything like that.
Yeah, that's a good point. So somebody asked, is there, I'm going to kind of reword, I'm sorry, Mary, I'm going to reword your question a little bit, but is there some sort of boot camp that people can look forward to, or education that after they get the results, it can sometimes be confusing, and we have live events like this, but what else can people do to help them understand the results, or maybe take some sort of boot camp or video series to understand?
There are a ton of events all over the country. It depends on how intense you want to get. There are your local genealogical societies are the best place to start. Many of them have DNA special interest groups. Then there are regional and state conferences regularly that have things. It's not just Roots Tech. There's networks of them. Then there's the institutes. For example, the Utah Genealogical Society has the Salt Lake Institute for Genealogy, SLIG, which is an intensive deal. There's a lot of things like that. We don't provide that because it requires a lot of bandwidth and manpower. Now we will be focusing more on providing more interesting and educational things over the next year. But as far as us doing one of those, it's probably not going to happen anytime soon. But there are plenty across the country.
And also linked on your dashboard, if you scroll down to the bottom under partner services, you'll see a button for your DNA guide. Diane Southard offers some great classes all the time. There's one I got an email about this morning. And so that's linked there. And I don't remember the details, but it's there for you.
She does. I know she does have a specific for Family Finder, specific for mtDNA, specific for YDNA. She has a lot for us. I do know that.
Yeah.
And if you're looking for more information on a specific topic, especially an advanced topic, I would recommend checking out our blog and our help center because some of the population geneticists and phylogeneticists, they will post some pretty advanced technical papers explaining sort of the science behind it. And me personally, I'm a little bit biased towards the help center because that's what I do with the company. But there are some articles in there as well on some of the concepts that we've been discussing.
Were you going to also say something, Janine? I think
I was just going to say that one of the members of one of the projects that I work with, he's over 80, took Diane's class in for YDNA and got a lot out of it. So you don't have to. There's no age limit on it. Let's put it that way. She's got some really great stuff.
And another thing that Leanne just posted in the comments, if you're looking for education on advanced topics, especially, the East Coast Genetic Genealogy Conference is great. It's been happening every year in the fall. We've all spoken there. It's a great time. Yeah.
It looks like we're coming up on the hour mark, and we've kind of gone through a lot of the broad questions. I do want to remind everyone that anybody who has submitted a very specific question about their kit or their matches, we will not answer that publicly. That is a question for our customer service team, so we would ask that you reach out to them. But we've gone through most of the broad questions, and so I think we're about ready. Is there anything that y'all would like to lead off with?
Yes.
Yes, Janine.
We are putting together a lot of really good material that will be in our virtual booth for Roots Tech. So when the virtual booth opens, please be sure to go to the expo hall, go to our virtual booth. There will be videos and download handouts. There's going to be all kinds of information there. So be sure to check that out as well.
And there'll be even more information at Roots Tech in person, because we always do a bunch of booth talks and everything. Yeah. So, and I'm really excited about all the cool stuff we've got planned for 2026.
Jim, any final parting words from you?
Just what they were saying about the Roots Tech virtual experience. Katy and Janine will both be in person. So they, if you're going to be there in person, definitely come up and say hi. I'm going to be hanging out in the virtual booth. We've talked a lot about the project administrators, and we always have some project administrator volunteers who help out to answer people's questions. So if there's a particular topic that you're just not sure about and you just really need some, you don't want to read an article or take a course, you want someone to actually talk to about it, feel free to pop into the booth. We have admins from all around the world at it, so it's staffed 24/7. So no matter where you are in the world, even if you can't make it in person, definitely check out the virtual experience.
And many times they're actually better versed in those things than we are because they do a lot of practical application. So it's even if you go to Roots Tech in person, you might want to take a few minutes on a break and jump on the chat to ask questions as well.
Mm-hmm.
Okay. Well, I think that's it for us. Thank you guys for joining. And thank you Roots Tech for letting us take over your social media. We're excited. We're to be Platinum sponsors, and we can't wait to see you guys in person and virtually from March 5th through the 7th. So we're excited. Thank you guys.
All right. Thank you.
Thanks, y'all.